Today's seminar is hosted by the National Cancer Institute RNA Biology Initiative. My name is Sandra Wolin, and I will be the moderator. We'll have two 20 to 25-minute talks, each followed by 5 to 10 minutes of questions and answers. So as each talk progresses, please write your questions in the chat, and I will ask them at the end of the talk. I also want to point out the link on this slide and in your email to Discord, which you can join after the talks end at 5 p.m. to meet the speakers and ask any questions you were unable to ask during the question and answers. Our first speaker is Astrid Haase, a Stadman investigator in the Laboratory of Cell and Molecular Biology at the National Institute of Diabetes and Digestive and Kidney Diseases in Bethesda, Maryland. Astrid carried out her Doctor of Medicine degree from the University, she earned her Doctor of Medicine degree from the University of Vienna in Austria, and her PhD with Wojtek Filipovich at the Friedrich Miescher Institute for Biomedical Research in Basel, where she studied microRNA and siRNA biogenesis and identified the protein TRBP as a partner of the dicer protein. She came to the United States for postdoctoral training with Greg Hannon at Cold Spring Harbor Laboratory, where she established the role of the zucchini protein as a nuclease that functions in piRNA biogenesis. In 2015, she established her own laboratory at the National Institutes of Health, where she has been studying the molecular mechanisms that underlie piRNA silencing. And with that, I'll give you Astrid.
Thank you so much, Sandy. Thank you for the kind introduction. And I would like to, and thank you everyone for being here and for listening to this. My lab looks at genomic conflict really from a small RNA perspective. What do we mean by this? When you look at your genome, that's actually not a peaceful place. 50% of our genomes, or sometimes even more, are comprised of mobile genetic elements or transposons, and they're disjunct fragments. And the potential activity of these elements, their ability to amplify and reinsert into different genomic locations, threatens genomic integrity and must be restrained. We are living with our transposons, and we are living well with them. How do we do this? Throughout evolution, our genomes have endured conflicts, and we've accumulated scars. And in very rare occasions, but important occasions, we have managed to co-opt transposon sequences for evolutionary innovation. But most importantly, we have learned to recognize and efficiently restrict transposons. So we silence them very early in development, in germ cells and in the early embryo, and keep them silenced throughout life. This phenomenon is broader because nucleic acids invade our genomes, and the defense of invading nucleic acids is universally conducted by RNA-guided immunity in all branches of life. In bacteria and archaea, mostly by CRISPR-Cas systems, and in eukaryotes, mainly by different RNA interference systems. What is common to these different molecular systems is that they rely on a rather short guide RNA to recognize non-self, or these mobile disinvaders, by base-pairing complementarity, and faithfully discriminate self from non-self. My lab studies PV-interacting small RNAs, or piRNAs, which are one of the 3 major conserved small RNA silencing pathways in animals. piRNAs mainly operate in germ cells, and they are essential for fertility, and thus for the continuation of a species. How do piRNAs meet the formidable challenge that transposon defense poses? piRNAs heavily rely on memory, and this memory is embodied in what we call piRNA clusters. And Julius Branneker famously called piRNA transposon graveyards. These are really large genomic regions in the genome that have a high density of disjunct transposon fragments. The piRNA system then transcribes this region into a long single-stranded RNA that really tries to capture antisense information of these transposable elements. The long precursor is exported in the cytoplasm, where it meets a processing site on the outer mitochondrial membrane and is chopped by a nucleus called zucchini PLD6 in a mechanism that pretty much looks like a food processor. So it's kind of chopped up in small fragments. And I would like to show you this in a little more detail. So this is one of the major pRNA generating regions in flies called flamenco. And what you see here is the beginning of the genomic cluster. And in gray depicted are all the pRNAs that we can observe associated with the PV protein in flies. So you see that this looks very chaotic and very messy. And we are facing millions of diverse fragments from these long precursors. Sorry, it doesn't, no, it advances. Coming back to the system, these small RNA fragments are incorporated into a PV protein, and this PV pRNA complex then heads out to survey the cell for the activity of transposons. If it encounters a transposon transcript in the nucleus, it recruits histone and DNA methyltransferases and establishes lasting epigenetic restriction. If it encounters a complementary transcript in the cytoplasm, it cleaves this transcript and results in degradation of this transcript. In germ cells, this cleavage mechanism can be coordinated by the germline-specific helicase basa or DDX4, and this coordination allows an amplification of pRNAs in the cytoplasm, which we call the ping-pong cycle. If you don't remember anything of pRNA mechanisms until so far, the most important thing is just this one complex. At the center of all of this phenomena resides the RNA-induced silencing complex, or PIRIS, which at its core really consists of one PV protein and its associated small RNA. And within this complex, the sequence of the pRNA determines target specificity by base pairing complementarity, while the associated PV partner determines the effector mechanism, resulting in transcriptional or post-transcriptional restriction. So the sequence of the small RNA is at the center of self-non-self discrimination. What we wanted to know in this project that I would like to tell you today is we wanted to know how do the millions of diverse pRNA sequences faithfully restrict all transposons while avoiding autoaggression. And precision is of the essence here, because if the system fails to restrict a single transposon, this can lead to enough activity and genomic damage that we lose germ cells and we have a sterile animal. But if it, on the other hand, mistargets a single essential gene, this could prevent further development of the embryo. To get at this question, we first actually had to look at how many pRNA sequences do we deal with? How big is a pRNA population? So we prepared PV pRNA libraries from ovarian somatic sheet cells, and these are the main data that I'm going to show you today. But we also integrated in our analysis beautiful recently published data from Haase lab that are million MIDI-bound pRNAs in mouse. So when you follow the pink line here, what we did is we looked at our data and we predicted a potentially saturated PV pRNA population. And what this saturation point shows you is that we predict a population of about 500 million molecules, that's the x-axis, with more than 10 million unique sequences on the y-axis. That's an ambitious expectation, but we have to face the biological limits. And the obvious biological limit is the number of pRNAs that a single cell can accommodate. To estimate this number, we use the spike-in sequencing approach, something very traditional. And Haase lab has done this too for mouse, and we are comparing this data here. So in purple, you see the data that we got for flies, and you see that we expect up to about 1 million pRNA molecules in a single cell in flies. And in mice, we expect between 1 and 10 million. So by all means, pRNAs are abundant. Their abundance is close to the abundance of ribosomes per cell. But there is a huge discrepancy between the expected size of the population and what a single cell can accommodate. What this tells us is that an individual cell can only accommodate a fraction, but never the entire sequence space of pRNAs. Next, of course, we wanted to know which pRNAs are in every cell and which ones generate cell-to-cell diversity. And to do this, we did something very simple. We just took all the sequences, lined them up on the x-axis, and counted their abundance on the y-axis. And what I'm going to indicate in the further slides for you is the threshold for a pRNA sequence to be present in every cell. So if it reaches more than 1 part per million, it could be present in every cell. If it's below that, it can definitely not be present in every cell. When we do this for PV pRNA data, so we line them up on the x and we look for abundance on the y-axis. When you follow me and look on the y-axis, what is immediately obvious is that pRNA sequences are very diverse in abundance, with 3 to 4 orders of magnitude in difference. And only a small fraction of pRNA sequences reaches an abundance that makes them abundant enough to be potentially present in every cell. while the vast majority of the sequence space is extremely low abundant and definitely not present in every cell. This explains something that all of us have observed for a long time. When you look at your piRNAs, and I see some piRNA people in the audience, and I look at my piRNAs, we will agree on the top most abundant sequences, but we won't agree on the entire piRNA sequence space. We can assay this. So what we did is we just did 3 biological replicates and we intersected them. So again, these are PV piRNAs. When we do this, shown in purple here, you see that the common fraction contains more than 80% of all the molecules that we observe. So this is pretty good reproducibility, but it only includes 13% of the sequence space of the diverse sequence. And these are the abundant sequences. While more than 70% of the diversity is actually extremely rare and only found in one or the other biological replicate. And together they only make up 8% of all the molecules. So from this, we conclude that while the piRNA sequence space is very large, we have only a few common sequences and many, many, many rare sequences. I've been working on small RNA biogenesis for my whole life. So of course we wanted to know is what makes a difference? Why are some piRNAs so extremely abundant and why are some so low abundant? What determines sequence abundance? And there were 2 things that we could easily test. We could ask, do they come from different precursors? And we could ask, are they made by the same biogenesis mechanisms? So first we looked at precursors. And what Pavel asked, very simply, is do abundant and rare piRNAs come from piRNA cluster transcripts? And when you look at the little insert, you see that about 50% of the common piRNAs come from these large piRNA clusters with a strong enrichment for this single flamenco cluster. To our surprise, most of the rare piRNAs, more than 90%, originated from these large clusters. What Pavel then did, he took the 450 top PV piRNA clusters and he lined them up on the x-axis, ranked by the mean abundance of the common piRNAs. What you can see, and I'm indicating the threshold here again, you can see that about the top 50 piRNA clusters produce piRNAs with a mean abundance that would allow them to be present in every cell with more than 1 part per million, while the rest of the clusters produce piRNAs that are not present in every cell. But the big surprise really came when we looked at the rare fraction, because all of these clusters produced rare piRNAs. And the sequence abundance between rare and common was between 1 and 2 orders of magnitude again. So what this told us is that 2 piRNA sequences that originate from the same long or potentially originate from the same long precursor molecule can be of vastly different abundance. And of course, we wanted to know why. How does that work? So we looked at known processing mechanisms. And the best characterized processing mechanism is the 1U preference of piRNAs, which has long been observed in flies and mice and other organisms. And we've recently contributed to the understanding by showing that this is actually established in a 2-step mechanism, first during biogenesis by the subprocessor, and then reinforced by selection during PV piRNA complex formation. What I'm showing you here is a metagene analysis of common and rare piRNAs. And the piRNAs that we observe are in the colored boxes, in purple and in teal. And the extended white region is the genomic region that in the extended interval. And then we are plotting the frequency of individual nucleotides across this genomic region on the y-axis as a fraction. And what you see is that both common and rare piRNAs show a preference to have a uracil in the first position. And they also show a small peak, 1 piRNA length downstream and 1 piRNA length upstream, likely indicating the previous and the next processed piRNA. They also show a very similar size profile. So we have reason to believe that they are made by the same processing mechanism. But when we wanted to compare them, we had to calculate the 1U bias. And we did this again, looking at individual clusters. So Pavel calculated the 1U bias as the observed or expected frequency for common and rare piRNAs for the 450 top piRNA clusters. And there the picture was pretty clear. piRNAs that are abundant and common usually have a stronger 1U bias than the low abundant rare neighbors, even when they originate from the same cluster. So this now I think poses an interesting hypothesis, and I don't have an answer for you here, but I would like to put this up for discussion. we have long been wondering, we know that there is this U bias, and we are getting more and more to know how it is established. But why? Why do you make this effort to establish this U bias? And one hypothesis that we would like to address is perhaps this bias during processing allows us to regulate sequence abundance and might allow us to enrich or augment piRNAs that work and actually eliminate or strongly reduce piRNAs that would have off targets, of course, in an evolutionary selection manner. We haven't been able to test this directly, of course, yet, but Dan did an interesting experiment. He just put all the piRNAs together again, just separated them by these 450 top piRNA producing clusters, and then he separated them by just the identity of their first nucleotide into one U, one C, one A, and one G piRNAs. And he asked, what is the relative sequence abundance? And when you look at this, the picture is pretty clear. If you and I are piRNAs and we come from the same precursor or the same cluster, if you have a U, you have a higher probability to be abundant than if you don't have a U. So 1U piRNAs generally seem to be more abundant than the 1C, 1E, 1A, and 1G neighbors. So far, in terms of mechanistic details, I hope I could convince you that for abundance, what really matters is the origin, the cluster of origin, and processing mechanisms. So we would predict that the top most abundant piRNAs should come from a top cluster and should have a U in the first position. And then the question is, of course, what are they and how do they function? So this was an easy experiment that Dan did. He just took the 1,000 top most abundant piRNA sequences out of these many millions that we see. And he looked at their 1U preference. And lo and behold, more than 90% have a U in the first position. More than 80% of all of the sequences come from a single cluster. And this is the only cluster that has long been known as a major transposon control region, and that is essential for transposon control, a known essential cluster in flies. And all of them regulate a single class of endogenous retroviruses, namely the gypsy class of endogenous viruses. And silencing these is actually well described for the flamenco mutant. So this was nice and really indicated that abundance would allow us to predict function. But we wanted to put this to the test. So we wanted to ask, how much does piRNA abundance predict function? So what Fenia then did, a grad student in the lab, she devised a simple sensor model. She took use of two fluorescent colors, red and green, and we are using red as a control here, and green as a sensor color. And what she did is she just brought the green color under the control of endogenous piRNAs. And she did this by adding a whole stretch with antisense complementarity to a piRNA producing region in the 3' UTR of the green color. And if she does this for a highly productive region of flamenco, she sees that endogenous piRNAs can silence the sensor. Her readout is nice because we can do it at single cell level. We just run all the cells through effects. And what you see in the flow diagrams below, when we do this for a control sensor, most cells, so each dot now represents a single cell, most cells express both colors and appear yellow. While when Tenia does this for her flamenco control sensor, she sees that 71% of the cells now are completely silenced of the green color and appear red in flow. And we take this fraction of completely silenced or red-only cells as a surrogate for silencing efficiency. Thea did a whole battery of sensors, sensing different stretches of flamenco and different other piRNA generating regions. And then she'd ask a very simple question. Is there a correlation between the silencing efficacy, the fraction of completely silenced cells, and the number of endogenous piRNAs that can target her sensors? And this is plotted here. So she's plotting the silencing efficacy on the x-axis and the number of piRNAs in parts per million on the y-axis. And what you can nicely see with the R-square is that we can explain more than 70% of the silencing efficiency just by the number of endogenous piRNAs that can target our sensor. So abundance really correlates with function in silencing. There's another point that I would like to draw your attention to, is that we don't know how high the threshold is, and I'm sure that biologically this will be regulated, But when you look what I indicated with the dashed black line is we have one sensor where we see 50% of the cells silenced. This sensor is targeted by close to 5,000 parts per million. So close to 5,000 piRNAs in a cell. So the threshold for complete silencing might be really high. And we wonder what this is telling us. With this, I would like to summarize our findings and allow me to have the first 2 slides for the younger audience, because there's one key point that I would like to convey in a story. Let's say this is the piRNA sequence space in 1D. Let me tell you a story. You're organizing a kiddie party. I have a 4-year-old, and we bring our 4-year-old to your party. If you tell our 4-year-old that these are the things that will be up for snacks, she's going to be your best friend. But you haven't told her yet that you don't have the same amount of each. And how her snack really looks like is like this. If you give her a bowl of fruit and then 1 candy, she might still like you. But if you don't have enough candy and you only give her fruit, she might not be your friend anymore. So going back to the data, what we found is that while the sequence space of piRNAs is humongous with millions of diverse sequences, there are only a few piRNA sequences that are highly abundant. Abundance in the system is regulated by the origin of the piRNA, so the cluster of origin, and within the cluster by processing preferences. And I hope I could also convince you that abundance correlates with function. What I want to leave you with is, first you might want to call them. So you could call the superabundant ones silencers, the medium abundant ones modifiers. And what might be interesting there is think about this is a mechanism that operates in germ cells. So if these medium abundant small RNAs converge on a target, they might contribute to reproductive variability in the system. But as sporadic piRNAs, this magnitude of low abundant things might never be meant to act. They definitely don't act in every cell. They're relatively sure that they don't act in every animal, but they might even not act in every generation. So what are they for? And the thought that I want to leave you with are actually these low abundant small RNAs, because we know that the system operates very well in steady state. But what we also know is that the piRNA system is able to include to accommodate new genomic invaders and get them under control by beautiful data from the Thurkov lab. And what we would like to put up for discussion is the idea that perhaps these extremely low abundant, non-functional piRNAs, or the mechanism that allows for their generation, provide a substrate for evolutionary tinkering and allow us to accommodate new genomic invaders over time. And with this, I want to thank my lab, especially Thenia, Dan, and Powell, who have done all the heavy lifting on this project. I would like to thank our collaborators on other projects, and of course, all the core facilities at the NIH and NIDDK Intramural Research Program for their very generous funding. And you for your attention, and I'm excited to take questions. Thank you very much. Sandy, do you want me to stop sharing?
Sure. So we already have some questions in the chat. So one is from Mariana Mandler, and she would like to know if you know if the 1U is modified.
No, that's a good question. No, I don't.
We have a question from David Clayton.
Wait, I don't think, because we have seen there are some really, really nice structures out there for other Argonaut proteins and the first base. And I don't think they've ever seen any modification.
Well, I guess I would wonder if they made those piRNAs in vitro or if they were native piRNAs then. Because if you start out with
You're right. The ones that saw the base were all loaded. Yeah. And for the others, we only saw backbone. Yeah.
So we have a question from David Clayton. Mm-hmm. Are the abundant piRNAs functional simply because they are most abundant or because of individual sequence?
Honestly, I think it's very, very simple. There's just a certain abundance threshold. I think the cool thing about piRNAs in contrast to microRNAs, this is an ancient system that is a defense system. So in a defense system, you really do what you can do. And when I started working on pRNAs, I thought the difficult part might be targeting all transposons. Honestly, now the older I get, the more I think the difficult part might be to avoid off targets, because you need a certain flexibility to include sequences. And I'm wondering, what is the mechanism that allows you to reduce something that could potentially be harmful if you are including a full antisense transposon? I think it's just a balance.
We have a question from Jin Li. I saw a high portion of others not from clusters in common pRNAs. Could you please comment on where they come from? And do you see different stability of pRNAs?
These are two very interesting questions. I can answer one of them. And I think our computational postdoc, Pavel, is in the audience. He's doing that now. In the original definition of pRNA clusters, there's a cutoff of 5 kb. And what we see is there's a significant amount of highly productive regions that is below this cutoff. So most of these things that don't define as a cluster by the original definition will just be slightly smaller. Yeah, around 3.5 kb. That's a very good question. In terms of stability and turnover, I have no clue. We know from tissue culture studies that if you knock down PV in ovarian somatic sheet cells, until you really lose function, it takes about a week. So the formed PV pRNA complex seems to be very stable. You don't see any small RNA fragments if you don't have a PV protein, and you lose the PV protein when you hit the biogenesis factor. So pRNAs and PV proteins are dependent on each other for stability.
We have a question from Tudlow asking if you studied the tRNA-derived quote unquote pRNAs, which are not from genomic clusters, but processed from tRNAs?
So there are a lot of pRNA sequences, and pRNAs are very ill-defined. They're literally only defined as PV interacting at the moment, in contrast to SIs or micros, where we know more about biogenesis to help define them. So what we do, and I think there's still a lot of cool questions about core pRNAs. So what we do when we get our data back, we eliminate everything that we can explain otherwise. So we actually first map against all abundant transcripts in the cell, including all tRNAs, and we eliminate this. We have done controls by just using beads and look what kind of fragments could also come as a background. And in our opinion, we can't be confident about these things. So we take all of this out. We haven't looked at this.
So we have a question from Leela Lackey. You mentioned avoiding off-target effects. Is there another mechanism for avoiding off-target binding or effects by pRNAs?
That's a very good question. The first part would be Alexia Ravin and Julius Brannecke when they were in Greg's lab, and afterwards they have shown nicely that you can add a sequence in a cluster, an artificial sequence, and it will produce pRNAs for you. So I think that would tell you that whatever is in a cluster will somehow end up as a pRNA in a PV protein. But there's a very nice paper from Kaja Vasic when she was a grad student in Greg's lab that shows that in mouse, there's a factor called RNF17. And if you take this factor out, which is a tutor protein, and if you take this factor out, the mice are sterile. And the first idea was perhaps it has no pRNAs, but that's not true. It actually has more pRNAs than it should have because it reactivates ping pong in adult male germ cells. So the idea that they discuss in this nice paper is that perhaps the reactivation and inclusion of more pRNAs than there should be result in autoaggression, and that causes this sterility. But that's something we know very little about.
So one more question from Jonathan Howard. Not familiar with pRNA modifications, but are all pRNAs from common to rare all still 2'-methylated?
So as far as we know, all pRNAs are 2'-methylated at the 3', yes.
And finally, we have a comment from Daniel Tennant saying he liked the 4-year-old explanation since he has a 5-year-old brain.
Thank you very much.
So we're going to move on, and I'm going to remind everyone to go to Discord at 5 p.m. if you have more questions. And there's, I think, one more question in the chat if you want to answer it there, Astrid. So I am delighted to introduce our second speaker, who is Jay Schneekloth, a senior investigator in the Chemical Biology Laboratory at the National Cancer Institute in Frederick, Maryland, and also a member of the steering committee of the National Cancer Institute RNA Biology Initiative. Jay received his undergraduate degree from Dartmouth College in New Hampshire. He then moved to Yale University, where he earned a PhD degree in chemistry with Craig Crews. As a graduate student, he developed the first cell-permeable PROTAC molecules. He then pursued postdoctoral training with Eric Sorensen at Princeton University, where he worked on the synthesis of analgesic natural products. And after a short stint back at Yale in the Center for Molecular Discovery, he joined NCI in 2011, where his laboratory uses synthetic chemistry and high throughput chemical biology approaches to develop chemical probes of RNA. And so with that, I'll give you Jay.
Thank you very much, Sandy, for the very kind introduction and the opportunity to talk here today. This is a really wonderful seminar series, so I'm definitely honored to be a part of it. So I wanted to talk to you today about the work that my lab does in targeting RNA with small molecules, and really what drove us to do this, and sort of how we think about this problem through the lens of a system that we like to, an RNA system that we like to study a lot. So what really got us interested in this topic is this what quite remarkable observation that if you consider the central dogma of molecular biology, we know of the 3 billion bases of the human genome, something like 85% of that information is transcribed into RNA, but just 3% of those sequences actually code for protein. Moreover, of the proteins in the human proteome, medicinal chemists have only been able to target or drug about 15% of them, which means that only a small fraction of the genome has really been targeted so far. And I think there's been a number of really key advances over the last 15 years, 20 years or so that have really enabled us to start thinking about this in a different way. One of this is thinking about the structure of RNA in a different way. Rather than thinking of an RNA as a largely flexible molecule that's devoid of secondary, tertiary, quaternary structure, the emergence of things like probing techniques like SHAPE and DMS map can give us insights into RNA secondary structure in large complex RNAs or even transcriptome wide. And also molecular techniques like NMR, cryo-EM, and XFEL now allow us to look at atomic resolution RNA structures and to understand how they bind to proteins or even small molecules. It's not only structure, but also an increased knowledge of the function of RNA, far from just coding for protein sequences or facilitating protein biogenesis. We know about all sorts of classes of non-coding RNAs, like Astrid just talked about piRNAs, but a variety of other classes of non-coding RNAs. And the biology that these non-coding RNAs regulate and its association with disease states, coupled with the potential for possibly targeting the mRNAs of undruggable proteins, really let us think about RNA as a therapeutic target. And if we can figure out how to do this, it would really be a transformative for developing new medicines. And so the question for us isn't why should we be doing this, but how could we not be doing this? And so if you want to think about targeting RNA with small molecules, there's a number of challenges here that are different for proteins. And our laboratory is really driven by 3 key questions. First of which is, what kind of chemical matter binds to RNA? Are you sort of constrained to the classical RNA binders, things like neomycin, that are highly cationic and bind to all sorts of RNAs inherently non-specifically? Or can you design or identify small molecules that have more drug-like properties and identify compounds that will bind more selectively to RNA? More than that, what type of RNA makes a suitable target for small molecules from a structural perspective? Frequently, we think about RNA structure in terms of its secondary structure as a defining element, but we of course know that RNA can fold into adopt really intricate 3-dimensional structures. And in fact, we know now that these structures often have hydrophobic pockets. And from a chemistry perspective, we think these 3-dimensional pockets are a really attractive way to think about what makes a good RNA target. But there is a disconnect between how medicinal chemists think about structure and how RNA biologists think about structure, that we think that that gap is beginning to be bridged. More than that, we think about what kind of tools are needed to understand RNA ligand interactions and how can we develop and harness chemistry to better understand this topic. So these might range from molecular simulations like molecular dynamic simulations that allow us to understand things like ligand dissociation, or designing chemical probes that we might use to map binding in a complex transcriptome and understand what is the selectivity you can achieve, or how broadly does a particular molecule bind to a particular group of RNAs. So I'm going to start by talking about the question of chemical matter, what types of chemical matter bind to RNA. So my laboratory has been using this technology called small molecule microarrays to look broadly and ask what types of small molecules bind to what types of RNA structure. The way this technology works is we use a robotic microarrayer to spatially array and covalently link tens of thousands of small molecules to a glass slide. You can then take your favorite RNA, label it with a fluorophore, and hybridize it to the glass surface. And then you can use a confocal imaging scanner to identify, to image your array slide and look for spots that increase in fluorescence corresponding to a binding event between your labeled RNA and the molecule that's printed in that location at the array. This turns out to be, my slide's not there. This turns out to be a quite powerful technology and pretty rapid. We can complete a screen in about a day if we're really industrious. Lab has now about 44,000 compounds that we can screen in this format. Requires low quantities of RNA, typically an amount that you can make in an in vitro transcription experiment in your lab, or if it's a particularly small RNA, you can even buy it labeled from a place like IDT or Dharmacon. This allows you to look at really important aspects. Not only does a compound from our library bind to the individual RNA of interest, but you can look at selectivity by comparing results of many screens. You can say, are there privileged classes of RNA structure that small molecules like to bind to, gaining insights into the targetability of a class, or are there even particularly molecules that have very, very highly selective interactions? We've now used this really broadly. We've looked at a total of something like 62 RNAs and 9 DNA structures. These are mostly nucleic acids involved in regulating gene expression in cancer and infectious disease, and really diverse structures ranging from 19 to all the way to 600 nucleotides, from a pre-microRNA hairpin all the way up to an entire UTR, for example. We now have a database of over 2 million interactions between small molecules and diverse RNA structures. And we've used this really broadly to publish on all sorts of different RNAs and small molecules. I'm obviously not going to go through this with you, but what I hope you'll take away from the information on this slide is just that the molecules that bind to RNA don't have to look like aminoglycosides, and they can really look like medicinal chemistry compounds that might bind to protein targets. And we think this is really compelling observation. So I'm going to spend the rest of the talk describing one particular RNA system that we find to be really useful and interesting. This is the pre-Q1 riboswitch. And we use this to sort of describe how we think about targeting RNA and how to study molecules that target RNA. So the pre-Q1 riboswitch, I won't introduce the concept of riboswitches to this audience, but this is an aptamer that's found in bacterial RNAs in the 5' UTR that binds to this small metabolite known as pre-Q1, illustrated here. And in the presence of the pre-Q1 molecule, the RNA undergoes a dramatic conformational change, forming this H-type pseudoknot, and the molecule binds at the center of that pseudoknot, harnessing a network of hydrogen bonding interactions to make a very tight specific interaction. The KD is around 2 nanomolar for this little compound. Now, a variety of different organisms, including commensal and pathogenic bacteria, have these riboswitches. It's a pretty small tractable aptamer, something that we can, even as a chemist, we could study in the lab. And one of the reasons we think this is really important is that this is sort of nature's solution to how to recognize small molecules with RNA. And so we've been studying this system for quite a while now. One of the questions we wanted to ask is, could we target the pre-Q1 riboswitch with small molecules? And so we designed a screening construct. You can see here we labeled the pre-Q1 riboswitch aptamer domain with Psi5 label and screened it against our library. And you can see here the result of one of the compounds that came out of this screen. And the compound we've identified is the structure shown here. It's illustrated in red. And you can see that it doesn't bind to basically any of the other 30 RNAs we've looked at. That includes other hairpins, triple helices, quadruplexes, and three-way junctions. It's kind of a surprise to us because this compound looks nothing like the cognate ligand pre-Q1 itself, other than the fact that it's kind of small and flat and has a pendant amine. But we were able to show that this compound binds to several pre-Q1 riboswitches from different organisms. The KD that we observe is around a micromolar, or a little bit less, around 500 nanomolar. We then, after we were able to show that this compound bound directly to the aptamer, we wanted to look more deeply at structure and function. And so we initiated a collaboration with my colleague at NHLBI, Adrian Ferri Diamare. And we put this compound into a functional assay. This is a single round transcriptional termination assay. And what you're looking at here is the full-length transcript and the terminated transcript. And what you can see is, as a function of the dose of compound, our compound is able to induce transcriptional termination. And in fact, it works the same way as the cognate ligand pre-Q1 itself. You can quantitate this as well if you perform replicates, and this works quite well. So we can show that not only does the compound bind to the aptamer, but it actually causes a functional consequence. Adrian's lab was also able to solve an X-ray co-crystal structure of the pre-Q1 riboswitch aptamer with our synthetic ligand. You can see here that the aptamer is in gray, and our compound sits right at the center of that pseudoknot. It's illustrated in cyan. This is very satisfying because this synthetic compound actually binds to the same hydrophobic pocket at the center of that pseudoknot that the cognate ligand binds to. Suggesting that diverse chemical matter that binds to this pocket can actually influence riboswitch function. It's sort of somewhat independent of the chemical structure. This is a relatively high resolution structure for RNA, 1.8 angstroms, and so we could get a lot of information from it. And we could say that although the contacts differ completely from the cognate ligand, the synthetic ligand occupies the same binding site. And you can see here that what drives this interaction is mostly stacking interactions and shape complementarity, as well as a single hydrogen bond with an A that's illustrated in this picture. One of the things that really excited us about this was the potential for structure-guided improvement. As you can see, there are a number of other residues that are nearby that we could perhaps capture interactions with to try and improve the quality or the affinity or the activity of this compound. And so we set out to do this. You'll see that although our compound had a pretty tight 500 nanomolar KD, it's a very weak inhibitor, and we had to go to much higher concentrations to see a functional effect. And what we did was we used computational modeling, where we could either dock or model different chemical scaffolds into that active site, or that binding site. And using this approach, we were able to design multiple different chemical scaffolds that we were able to experimentally demonstrate actually do bind, although they differ slightly from this initial construct, or sorry, from this initial chemical scaffold. And in fact, several of these compounds display dramatically improved activity in the biochemical assays, despite the fact that most of their affinities were about a micromolar. So it's about the same affinity for the RNA, but drastically improved activity in the functional assays. So I'm going to show you an example of one of these compounds here. You can see that we've synthesized a compound that contains a xanthone scaffold rather than the dibenzofuran scaffold. And when we modeled this compound in, we predicted that this carbonyl would make a second contact to the RNA. And in fact, Tom Numata, who was a former postdoc of Adrian's, who's now working in his own lab, is able to solve a co-crystal structure of this compound. And it bound exactly as we had predicted, modeling. And in fact, we're able to observe that there is likely a contact with this 2' hydroxyl here. And so very satisfying that the experiment actually validated what our computational predictions suggested, and we were able to use this rational design to improve the properties of our molecule. And more important than that, it's really not the KD itself, but actually the nature of the interaction that impacts the activity of the compound in these assays. And so we've been continuing to study this and understand better how these compounds binding to this aptamer influence the switching activity. Okay, so so far I've shown you structural techniques and biophysical techniques, but of course we all know that RNA is a dynamic molecule. And we really wanted to gain a sense as to whether or not these different molecules were sort of differentially impacting the flexibility and dynamics of the RNA itself. And so to answer this question, we initiated a collaboration with Pratush Tiwari and his group at the University of Maryland. And Pratush is a theoretical chemist, and he's developed a new series, a new way to do molecular dynamics called RAVE, which is an enhanced Bayesian sampling technique for molecular dynamics. And what it allows you to do is you can simulate, this is an all atom simulation, but uses an AI assisted technique to do very long timescale molecular dynamics simulations. And so, for example, we can do simulations of the pre-Q1 riboswitch aptamer in the presence of the cognate ligand. As you can see here, cognate ligand is in red. And you can also see here in the presence of our synthetic ligand, which is illustrated in green. And these simulations really did illustrate what we thought, which is that you can really see some pretty substantial differences between the cognate and synthetic ligands. Those differences primarily lie in the flexibility of the lateral loop and the stem that's below the binding site, illustrated by these two red arrows. And we can see that overall, the cognate ligand does a much better job of just stabilizing the overall RNA structure. Okay. And so this turns out to be a really powerful technique to look at RNA flexibility and understand how these molecules bind. Another way in which we can use this is because it's an AI-assisted simulation, we can look at events that occur in a timescale that's much longer than you typically might see in an MD simulation. In this case, we took a look at ligand dissociation pathways, and we were able to actually simulate how the two molecules differentially dissociate from the RNA itself. And so this also allowed us to get some insights into specific bases that are important for allowing the different molecules to dissociate. One of the important things about carrying out these MD simulations is, do they actually correlate with experiment? And so what we did was we performed shape map experiments on the pre-Q1 riboswitch. This is using the 2A3 shape reagent that was recently published by Danny Incarnato's lab in the Netherlands. And here you can see the individual nucleotide shape reactivities, which correlates to the flexibility of an individual nucleotide. This is the pre-Q1 aptamer here, and then these are just adapter sequences that are not relevant. We were really pleased to see that when we looked at the individual nucleotide flexibilities that we observed in shape experimentally, coupled with the C2C2 distances that we measured in the simulations, we see excellent correlation. So not only can we simulate these complex RNA molecules, but we can really understand how ligand binding influences nucleotide flexibility. Okay, so far I've talked to you about a sort of in-depth understanding of a single RNA, but if we really want to think about targeting RNA, we need to think more broadly about how molecules interact with diverse collections of RNA. And so here we turn to one of the chemical biologists' favorite tricks, which is to do photo cross-linking and pull-down assays. Here we designed and synthesized a variety of what's known as clickable photo affinity probes, where we took the pre-Q1 molecule and coupled on a diazurine as well as an alkyne. And what this allowed us to do is, if you photolize, if you irradiate that diazurine, it'll form an alkyl diazo group, which will react with nucleophiles in the nearby space, such as the RNA that it's proximal to. So we synthesized a variety of these probes, and we were able to show that the nature of the probe, the side chain and the cross-linking, had a dramatic effect on the cross-linking efficiency. I'm not going to go through in detail here because of the constraints of time, but we were able to show that as a function of probe concentration, we could see a dose-dependent increase in photo cross-linking efficiency, either in the mass spec or in gel-based analyses. We also solved, along with Tom Numata again, a co-crystal, X-ray co-crystal structure of our probe bound to the pre-Q1 aptamer, and we could show that it binds the same way as the unmodified probe. And in fact, there are several bases that are very near to that electrophilic site. And we could show that this probe is actually still functional in transcriptional termination assays. We could also show that you could use this probe to do target engagement studies. So you can compete away photo cross-linking events with unlabeled pre-Q1. That's shown here in this top gel. We could also show that the addition of a competitor RNA had no negative effect on cross-linking. So if you add in XST RNA as a competitor, there is no competition because the probe doesn't bind to that RNA at all. We next took another step forward, and we took total human RNA extracts, total RNA, and photo cross-linked our probe to that, clicked on a dye and ran it out on a gel. And as you can see here, this is a complex experiment, but what you can see very clearly here is that when we dope in the pre-Q1 aptamer into human total RNA, you can see cross-linking here to the aptamer, as illustrated here in this fluorescence gel, but we see essentially no other cross-linking. What that means is that this is really an exceptional cross-linking event, or exceptionally selective cross-linking event. We really wanted to do this quantitatively, and so we did a pull down and sequence the RNA that came out. This is a technique known as ChemClip, or sometimes it's referred to as PearlSeq. It's been used by others like Matt Disney and industry groups, and also Pete Schultz and his group have used these approaches. So what we see is a significant increase or enrichment of the pre-Q1 riboswitch aptamer. And if we compare that in a volcano plot to all other RNAs that we're looking at, you can see that it's just really essentially the only RNA that's enriched. So when we photo cross-link and pull down, it's the only RNA that we see. Another way to look at this would be through an identity plot. And again, you can see the only RNA off this identity is the pre-Q1 aptamer. So it's an exceptionally selective cross-linking event and binding event. What was really interesting, though, is to ask, well, what if there are aptamers for pre-Q1 within the human transcriptome? And so we performed the same experiment in the absence of exogenous bacterial aptamer. And here we identified a number of different transcripts that were enriched by this photo cross-linking probe. And in fact, we see that several of these probes not only can be enriched, but they can be competed away with unlabeled pre-Q1, meaning that this is a specific interaction. We see a total of 16 different transcripts that are enriched. These include several different types of RNAs, including histone mRNAs, as well as TERK, which is the telomerase non-coding RNA. Intriguingly, all these RNAs are known to contain pseudonuts, but we don't yet know where the compound is binding on these complex RNAs. Okay, so with that, I'm going to just sort of sum up and talk about why we think this is really important. And here, hopefully I've convinced you today that drug-like small molecules can bind to and modulate RNA function by engaging three-dimensional structure, such as I showed you with the pre-Q1 riboswitch. We use this microarray-based technology to enable rapid ligand identification and understand targetability. The principles of molecular recognition for RNA are in many ways analogous to proteins. You can design and improve inhibitors, and atomic resolution structure, at least for us, was key to really making several key advances. We use this photo cross-linking to enable prospective metabolite binding sites transcriptome-wide and characterize the selectivity, and using this type of photo cross-linking probe, which turns out to be quite valuable. So from here, where are we going? Well, we'd like to improve the potency of our synthetic inhibitors to see cellular activity. We think this is a very powerful potential way to look for empirical riboswitch discovery in other bacteria, which would really complement the primarily informatics-driven approaches that have been used to discover riboswitches so far. And something that we think is really exciting, which would be to look for aptamers of metabolites in human transcripts, which could be quite interesting as regulatory sequences or just drug binding sites potentially. So I'm going to stop here, and I'd like to thank the lab. I'm really fortunate to have a just absolutely spectacular group of postdocs and postbacs who work in the lab. I want to especially thank Colleen Connolly, who's now left the lab, but also Shafali Parmar, Samirtha Balaratnam, and Curran Rhodes, who did the work that we talked about today. Our collaborators, Pratush and Adrian and Danny, who I mentioned throughout the talk. Thank you again, Sandy, for inviting me, and I'd be happy to take any questions if there's any time. I know that I went a minute or two over. But thank you very much.
So thank you, Jay. You have a bunch of questions. I'll remind everyone that if we don't finish them, you can ask them in Discord at 5 p.m. So first question is from Lorenzo Baranti. Are the compounds in the small molecule array covalently bound bonded to the slide? If yes, do you have different replicates where you change the attachment point to avoid false negatives? And then finally, what is the affinity range the assay is sensitive to?
So a great question. I get this question a lot. They are covalently modified. They are covalently attached to the slide. Every molecule in the library has an amine or alcohol that bonds to an isocyanate or epoxide surface on the slide. At least that's how we think it's bonding. It's very hard to characterize 40,000 different chemical attachment points. We do not have a way in this case to look for false negative controls. And we do know that we are probably missing at least some things. But it's very, it's very hard to sort of control. We've toyed with looking at photo cross-linking where you can stochastically label the small molecule to the surface, but haven't yet been able to make that work as well as the isocyanate surface. And so most of our experiments are done using that, the label, the isocyanate, where it's labeled directly through an amine. And what was the third part of that question, Sandy, again?
I think the affinity range of the
assay. So the affinity range, we typically see, we would call it hit validated if it has an affinity range at the high end of like 2-digit micromolar, so around 50 micromolar. But we also see things that are much lower. Frequently, we see things that are much more like 1 micromolar. And in a variety of cases, we have now nanomolar binders for a variety of different RNA structures. So commonly, I would say it's actually about what you would see for a protein if you weren't using a biased library for just a random protein target. Yeah.
We have a question from Fabrice Leclerc. What would be the kind of small molecules to screen against RNA targets? For example, kinase inhibitors or fragments of these?
So our library consists of not an RNA-focused library. This is just a medchem library. So it's meant to be comprised of compounds that represent drug-like chemical space. And so it's not anything special. Now, what we've learned from screening all these targets, we're now beginning to make an RNA-focused library. But this is not, at the outset, we didn't know. And so this is just a drug-like chemical library. We do also have FDA-approved drugs in the library, and we also have some fragments. But we haven't really published much on that yet, but we're planning to in the future. But they're really just medchem compounds.
You have a question from Andy Saltz asking if you've compared your hits from your fluorescent screens with data from Matt Disney's Inforna approach, and whether you identify any of the same compounds.
So I don't know all of the compounds that we're obviously really familiar with Matt's really excellent work. I don't know what, I don't know how all the compounds that Matt has. One of the features that Matt has identified is that things like benzimidazoles are sort of like privileged scaffolds for RNA binding. And in our cheminformatic analysis of our hits, we also see that. So there are definitely commonalities between what we see and what Matt sees. We identify other things as well, but yeah, there is some agreement between what we see and what Matt sees for sure.
We have a question from Anna Krichevsky. Do you envision small molecules not only as inhibitors, but also activators of RNA function, such as microRNAs?
I would love to see that. And, there's so many different ways you can go about it. But yes, we would love to see that. You can imagine things that make better translation, right? That are sort of potentiators of translation or activators of microRNAs. I would love to see that. And I think we just need to find the right systems to interrogate for that. But yes, that's something we think about a lot. Yeah.
We have a question from Bon Trin. Can you elaborate on your chosen conditions for binding array screening, such as temperature, pH, and salt, since these can affect structure and binding?
They do, and we've interrogated a lot of those variables. We frequently do our screening at room temperature to 37, depending on the RNA we've looked at. And in cases of simple RNAs like hairpins or quadruplexes, we know that the fold doesn't change much with under the right conditions, and we try to spectroscopically validate the proper folding of the RNA if we can. For larger, more complex RNAs, you can do things like screen the RNA in the presence of high or low magnesium to sort of unfold the RNA, and in that case we absolutely see drastically different results, right? So very different classes of molecules will bind like a folded versus an unfolded RNA. So it's very important to get the fold correct.
Okay, so I think I'm going to stop here because it's 5 o'clock and remind everyone to go to Discord and ask if they have more questions to ask. And lastly, I want to give a plug for the next seminar in the RNA Collaborative Seminar Series. It will be held October 6, 2021 at 9 a.m. Eastern Standard Time. And it is hosted by the National University of Singapore Cancer Science Institute, Singapore RNA Biology Center. So that's October 6th, 9 a.m. Eastern Standard Time.
Thank you.
And I'd like to thank both our speakers, Jay and Astrid, for great talks.
Thank you, everyone, for coming again.
Thank
And
you,
thanks for the
Sandy.
invitation.
Yes, and thanks to all of you. It was a great turnout.
We'll see you on the other side. If you
I'll
have
see
more
you in
questions.
Discord. Yeah.